This is great. It took 11 chapters, but we're finally starting to talk about DNA. So we're finally ready to start talking about how do we actually get the things that we see in heredity? Like what is actually happening underneath the hood? Why do we see the patterns that we are able to observe? And so this is my favorite part of genetics. Up until now, we've been kind of talking about these classic experiments and overall trends with heredity, like with Punnett squares and dominant recessive stuff like that. But now we're getting into like how do these things actually happen? What is happening inside of the cell? And so overall, this is now molecular genetics. So this is the study of DNA structure, the function at the molecular level, what is actually happening in terms of interactions between proteins and functional units within the cell. And molecular genetics is what we would call that branch of biology. Everything that we know about genetics comes from our understanding of DNA, how it interacts with RNA, and genetic material overall has to do four things. And what we're about to talk about is the discovery of DNA, the structure of DNA, and that DNA actually does anything inside the cell. And so uh, the question for this first part is what is the molecule that actually is transmitting information from one cell to the next as cells divide? So when you get a new generation of life, how is information transmitted from one generation to the next? So right now we all know that that's DNA, but we're about to talk about how did we discover that? How did we know that it was DNA and not some other molecule? And so the things that we knew is that whatever the molecule is, that transmits this information from one generation to the next, first of all, has to contain information. Like it's got to be able to code for information. That information has to be able to be transmitted. We've got to pass it from one parent to the offspring. We have to be able to replicate it because we know that whenever one cell transmits information to another cell, the original cell still has information. And so the information must have been duplicated at some point. And then finally, there needs to be variation. So when we look at that information, that molecule that has that information, there's got to be variation in the molecule so that that way you can have diversity. But at the same time, it can't be so different that now you have a totally different molecule that isn't going to be compatible as we go from one generation to the next. And so at the very beginning of this whole story, we're going to talk about three experiments. But these three experiments are going to let us know that DNA is the molecule that is doing these four things. So at the beginning, people knew that cells are made of the four macromolecules. So remember, the four macromolecules are carbohydrates, lipids, proteins, nucleic acids. People already knew, just chemically, cells are made of those four things: proteins, lipids, carbohydrates, nucleic acids. But they didn't know which of those four molecules is the one that transmits information from one cell to the next. The running idea, the running hypothesis at the time was that it was probably proteins, that the proteins inside of a cell are used to transmit information to the next generation about what proteins it's going to have. And that isn't a crazy idea. Inside of the cell, proteins do most everything. Proteins are the enzymes that carry out most all chemical reactions. And when you're looking at one cell to another one, from one species to the next, that's really the thing that changes from one cell to the next, or the proteins that you see in the cell. So it does make sense that like proteins would have a role here. And everyone knew that DNA was inside of cells. You could see that all cells had DNA inside of them, but people didn't really know what they did. It wasn't really know what the function of DNA was. It was thought maybe it was some kind of like linker molecule or something, but that it was not transmitting all the information. And so this diverse experiment we're going to talk about, it's Griffith, Frederick Griffith. This is in the 1920s, and he's trying to understand how pneumonia works. And so he's got this uh, organism, Streptococcus pneumoniae. And so this is what causes pneumonia. And he's got two different versions of pneumonia. He's got a rough strain, the R strain, and he's got a smooth strain, the S strain. Well, the rough strain doesn't actually kill a mouse. So if you have a mouse and you inject it with R strain pneumonia, the mouse will survive. It will not die. That's what this is showing, is that we got rough strain living, rough strain bacteria. We inject it into a mouse, mouse survives. But if you take the S strain bacteria and you inject that one into the mouse, it will die. So S strain pneumonia kills a mouse. R strain pneumonia does not kill a mouse. And remember, this is a bacterium. And so that's why it has those names, is that the culture itself will look either rough or smooth when it's grown out on a plate. And the smooth one is the one that is virulent, that will kill the mouse. Okay, so that's what these first two experiments are. Okay, now he starts to kind of manipulate stuff. And so he's going to have heat killed bacteria. So remember that when you expose bacteria to heat, it will kill them. But the reason that it is killing them is because the heat will denature proteins. And so in this instance, the manipulation he's doing is just exposing the pathogen to heat. And he knows that if you expose it to heat, it's going to destroy the proteins inside of that pathogen. Okay, so he's going to take heat killed S bacteria. Okay, so heat killed S bacteria, injects that into the mouse, and it survives. Remember, S is supposed to kill the mouse, but in this case, if you heat kill S first, it will not kill the mouse. This experiment isn't really that surprising, right? Like you killed the pathogen before you inject it into the mouse. So when the mouse survives, it just means that the pathogen wasn't effective anymore because it was already dead. And actually, that also helps to support the idea that the proteins are the thing that is important because we know that the proteins are what is destroyed when we do that heat kill treatment. Okay, now we get to the final experiment, and this is the one that is really interesting, is we're going to have live type R. So remember, R strain pneumonia should not kill the mouse, and it's alive. And we're going to, again, get heat killed S type pneumonia. Well, remember, heat killed S type pneumonia by itself didn't do anything to the mouse. The mouse survived. Live type R also did not do anything to the mouse. The mouse survived. But when we mix the two of those things together and then we inject it into the mouse died. And so that is unusual. If R by itself doesn't kill a mouse and heat killed S by itself doesn't kill a mouse, why is it that when you mix the two together that somehow it kills a mouse? And this is a repeatable sort of a outcome. He would do it over and over again and keep getting the same result. And so the conclusion is that somehow information from that S strain bacteria, it was heat killed, but somehow information from that S strain is being transmitted over to the living R strain, and then the R strain is killing the mouse. That somehow the R strain is learning something from the S, and that is what's enabling the R to kill the mouse. And so he didn't know what the molecule was. He didn't know how that information is being transmitted. But what it does indicate is that whatever that molecule is that's transmitting the information must not be proteins. Because when you do the heat kill treatment, you're going to denature the proteins. And so whatever is transmitting that information from the S over to the R must not be a protein because the proteins would not have been intact after that heat kill treatment. And so he doesn't know what it was, but he's going to say that there's this thing called transformation that's happening. And somehow bacteria can incorporate information from other bacteria, even if they're dead. And that's called transformation. And that phrase, transformation, you'll notice that persists until today. And so when we talk about bacteria, that they can incorporate plasmids and then express those genes after incorporating them from the environment, we still call that transformation. And that's exactly what was happening in this experiment. And so we had two important outcomes from this. First is the discovery that bacteria can transform. And the second is that whenever that transformation happens, it must not be with proteins. But again, it doesn't know what it is. Now we fast forward another 20 years. Now we're with the next group. So this is Avery, McLeod, and McCarty. I used to say MacLeod, but I had someone who was in class one time, a student who was from Scotland, and he said that's pronounced McLeod. Avery, McLeod, and McCarty. So that's how you say that. All right. So 1940s. They've got the same kind of like model here. They've got the type R pneumonia, they got the type S pneumonia. The thing that they have that is different is by this point, a centrifuge has been developed, like that exists. It's been invented. Remember, a centrifuge is that thing that's in a laboratory where you can spin stuff really, really quickly. And when you spin them really, really quickly inside of a test tube, things are going to separate out in the test tube according to layers. The heaviest stuff's going to go to the bottom, the lightest stuff is going to go to the top. And so the reason why this is helpful in this experiment is that means that they can separate out nucleic acids from proteins. Proteins are big and heavy, nucleic acids are smaller and lighter. And so when they do basically the exact same experiment that they did over here, where we're going to mix these heat killed and these living strains, they aren't going to mix all of the heat killed S. They're going to be able to separate out the proteins from the DNA, from the RNA, from the carbohydrates, and add only one of those things at a time. So if we look at their individual experiments, here's type R cells, right? And if they take type R cells and they put them out on a plate, type R cannot grow by themselves. In this experiment, type S can grow on the plate, but type R cannot. And so if they have type R by itself, it can't grow. But if they mix the type R with extract from the type S will transform the R, and now it can grow on the plate, kind of like when the type R was able to kill the mouse. Okay, so they know that there's information inside of this type S, and if they put that type S in with the R, all of a sudden the R can grow. Okay, so now they have these centrifuges, but they also have these enzymes, and its selective enzymes they have are DNases, RNases, and proteases. So remember, the way that we name enzymes is the name of the enzyme is going to end in ace. That lets us know it's an enzyme. And then whatever that enzyme acts on, whatever it breaks it down, will be the first part of the name. So a protease is an enzyme that breaks down proteins. An RNase is an enzyme that breaks down RNA, and a DNase is an enzyme that breaks down DNA. So here they're adding DNase. So it's still the extract, but all the DNA has been destroyed. And so this extract would have proteins inside of it. And when they mix that with the type R, it's not able to transform. Transformation doesn't work if you destroy the DNA. Now they do the same thing with RNase. So they're going to destroy the RNA, but the proteins and the DNA are intact. And transformation does work. Okay, so it's either DNA or proteins that are helping for the transformation. Now, if we do protease, that means the proteins are destroyed, but the DNA and the RNA is intact. It does transform. And so you might be, okay, well, then both of these transformation happened, right? Yes. But transformation happened, but the thing that was in common with these two slurries, the thing that wasn't destroyed, was DNA. And if you do destroy DNA, transformation doesn't work anymore. So this shows that DNA is necessary in order for transformation to happen. Like it doesn't work unless DNA is there. So you might have been like, okay, well then this seems like that's good, right? Like it has to be the molecule that's responsible for heredity. And this is very strong evidence. The issue is just that this is kind of like a negative control or like negative evidence, and that we know that DNA must be present, but we don't actually have any evidence to show actively that DNA is the thing that's allowing for heredity to occur. We're kind of saying like process of elimination, like it can't be this, can't be that, so it must be this, which is useful, but it can't be our final answer. We need something even more definitive to prove that DNA is the molecule of heredity, because still there were plenty of people who still thought it was proteins who are transmitting information from one cell to the next. And really, truthfully, proteins can transmit some stuff, a lot of stuff from one cell to the next. It's not the overall hereditary molecule, but proteins can perform jobs inside of different cells. Okay, now we go to the next, the third experiment, Hershey-Chase experiments. This is the 1950s. So again, Griffith was 1920s, Avery, McLeod, and McCarty 1940s. Now we're in the 1950s. And so Hershey and Chase, they have got an even more sophisticated experiment here. They're not going to use the pneumonia anymore. They've got a different model. And they've got this bacteriophage. And remember that word, bacteriophage. So bacteriophages are viruses that infect bacteria. Okay. So the phage they're working with is called T2. So in this picture, this is T2, and it infects E. coli. So in this picture, here's the E. coli, right? So they got a virus, they got a bacterium, the virus will affect the bacterium. Okay, well, what they're going to do is they're going to grow two different strains of virus. They have one strain of virus that has got radio-labeled or radioactive atoms of sulfur inside of it. All of these have got sulfur that is radioactive inside of them. And the reason that's important is because proteins can have sulfur inside of them. Not all proteins, but many proteins have sulfur inside of them. But DNA does not. And so that means that all of the proteins are going to be labeled with radioactive sulfur in this group. And then in the next group, they've got viruses that were grown in an environment that has radioactive phosphorus. It's got radio labeled phosphorus or radioactive phosphorus. Phosphorus is found inside of DNA, but phosphorus is not found in proteins. And so with these two models, they've got viruses, T2 viruses that have radio-labeled phosphorus, which means they have radioactive DNA. And then they've got another strain of viruses that have got radio-labeled sulfur, which means they've got radioactive proteins. So now they let those two strains, those two viruses are going to infect E. coli. And after they do that, they take the infected E. coli's, they're going to centrifuge them up, and they're going to look to see, okay, where's the radiation? And what they find is that these E. coli cells never had any sort of isotopes inside of them. So the interior of those E. coli does not have radiation inside of it. They're not labeled. They're just normal E. coli cells. And again, those are the ones that had the radioactive proteins. These cells that were grown with the radioactive phosphorus, they are radioactive, these E. coli cells. They are radioactive. They got radio labels inside of them, and they're glowing. And so what that tells us is that the thing that was injected into these E. coli cells was the DNA. The thing that was not injected into these E. coli cells was the protein. And so this was like our positive, active evidence to show us that DNA is the genetic material, because it is the thing that is injected from one cell to the next, and it must be sufficient to allow for heredity, and that those E. coli cells were now infected with the virus, and so they're going to now express the viral information. And so this was our third experiment, and that is how we know that DNA is the molecule that's used to transmit information from one cell to the next and is responsible for heredity, is this very last one.